Hi there. Today we're diving into the fascinating world of antisense oligonucleotides, or ASOs for short. These are short synthetic strands of nucleic acids designed to bind specifically to RNA molecules. By binding to their target RNA, ASOs can modulate gene expression and treat various genetic disorders. ASOs are short synthetic strands of nucleotides. Essentially, they're designed to be the opposite, or antisense, of a specific mRNA sequence in our cells. ASOs come in different categories based on how they work, their mechanism of action, and how they are designed. Let's explore the main types currently in research and in development by academia and industry. First, we have steric blockers. These ASOs bind to mRNA and physically block translation or other processes without degrading the mRNA. These sequences are carefully selected to match the target mRNA exactly, ensuring precise interaction. When steric blockers bind to their target mRNA, they do so by forming Watson-Crick base pairs. This binding is highly specific due to the complementary nucleotide sequences. Now, let's talk about what happens next. Once the steric blocker is bound to the mRNA, it acts as a physical barrier. This means it can obstruct various molecular processes that would normally occur. For instance, steric blockers can prevent ribosomes from binding to the mRNA, effectively blocking translation. No translation means no protein synthesis from that mRNA template. Steric blockers can also interfere with other critical sites on the mRNA. They can block splice sites, which are regions where the mRNA is cut and joined to form mature mRNA. By doing this, they can alter the splicing pattern and potentially create different protein variants. They can also block microRNA binding sites. MicroRNAs are small RNA molecules that regulate gene expression. By preventing microRNAs from binding, steric blockers can modulate the stability and translation of the mRNA. To sum it up, steric blockers work by binding to mRNA and acting as a roadblock, obstructing various molecular processes. They don't degrade the mRNA. Instead, they prevent it from being used or modified in the usual ways. This mechanism has exciting therapeutic potential. For example, in diseases caused by overproduction of harmful proteins, steric blockers can be designed to specifically block the mRNA of those proteins, reducing their production and alleviating disease symptoms. If you are interested in learning more about antisense oligonucleotides, synthetic backbone and nucleobase chemical modifications, or you work with ASOs and are looking for good, reliable manufacturers as one of your suppliers, consider getting in touch with us on our Biotechnology Reviews journal, available on LinkedIn and other social media sources. We also have excellent in-depth articles covering all aspects of ASO design and mechanisms on our LinkedIn newsletter. Stay curious, stay innovative. I'm Luke McLaughlin. Thank you.